Can you start the session, please?
Oh, okay.
Thank you, Prophet.
Actually, I try to answer my own exam. Try to help me to prepare the review for exam. Just a second. 30 seconds. Could you wait 30 seconds? Here. I try to answer my last question. Yeah, I'm ready. Okay, let's start to make review for midterm exam. I made 10 questions, and for each, you can choose 1 out of 5. So it will be the multiple choice. So shouldn't be very difficult. So please let me know if those are too easy. So I try to make some more difficult question for final exam. Okay, so the question number 1. I asked about the raw materials, ingredient to make sake. So we have 4 ingredients, major ingredient to make sake. So this is a slide. I have not. I need to share. Sorry. One more time. Share Windows. Review. So you can see the slide.
Yes, Professor.
Yeah, okay. So 4 major ingredients for making sake are water, yeast, and koji rice, and steamed rice or rice. So we can make sake, rice, koji rice, and yeast, and water. Just we need to mix these 4 ingredients to make sake. Next, rice. We usually use Japonica rice to make sake, not Indica, not Japonica. And especially non-glutinous Japonica rice we use to make sake. We can make sake by using glutinous rice of Japonica, but it is not very popular. Most of sake were produced by using non-glutinous rice of Japonica rice. So these are a requirement of sake rice for sake production. 1, we prefer to use rice with large size of shinpaku, white core. This is very important. With strong character, it means shinpaku shouldn't be broken by polishing. So this is the one requirement for making sake. Next, sake rice shouldn't be sticky after steaming. Number 3, sake rice need to be easily allow fungi to be penetrated. So fungi can penetrate easily into inside of rice. So this is the 3rd requirement for making sake. And 4th requirement of sake rice to make sake is rice can be dissolved easily. Last requirement of sake rice is low iron, low protein content. So sake rice shouldn't have much iron and protein. You don't memorize all 5 requirements. I added, other requirements. So you need to choose one inappropriate answer. These 5 are, major requirements for making sake regarding as the ingredient of rice. And when we polish rice grain and getting smaller and smaller by polishing, and we can discard fat very efficiently by polishing, and we also can discard mineral and protein. I made a long answer. And if we make polish rice grain by polishing rate of 90%, like this, and we can discard fat very much, about more than 70% of fat will be discarded by making rice grain polishing rate of 90%. Protein, only 10% of protein can be discarded by polishing rate of 90. So fat can be discarded very efficiently by polishing, the polishing rate of 90%. So I made some question regarding as this slide. So you can memorize. We can make classify sake with or without addition of distilled alcohol. When we do not add any distilled alcohol, and we can put the name word of junmai. It means pure rice on the label of sake. So if you find the word of junmai on the label of sake, and you can find that the sake does not have any distilled alcohol. So if you will be asked the word on the label of sake, which does not have any distilled alcohol, and you can find this word, junmai. Jun means pure. Mai means rice. It means sake does not have any distilled alcohol. And next question, if you have sake which was made by using rice at the polishing rate of 70%, 70%. So sake shouldn't, producer or sake maker shouldn't use these words. If sake was made by using 70% polishing rate of rice, either honjozo or junmai, simply junmai sake. Yeah, right. If sake made by either rice at the polishing rate of 70% with distilled alcohol, and it will be called as honjozo. And if sake was made by using rice at the polishing rate of 70% without any alcohol, and we can call it as junmai sake. But we cannot use ginjo or daiginjo. We cannot use these terms, these words, if sake was made by using rice at the polishing rate of 70%. And if we have sake made by rice at the polishing rate of 60% with distilled alcohol, and it would be ginjo. We cannot put the word of junmai. And sake made by rice at the polishing rate of 60%, we cannot use the word dai ginjo. Dai means large, big ginjo, great ginjo. And ginjo, just simply ginjo. If we have sake, the polishing rate of 60% rice was used. And we cannot use this word. We can use only ginjo, this word. And you also learn some history of sake production at the temple in Nara Prefecture in between 1336 and 1573. This temple developed 5 major technique to make sake. One, and you may remember, they began to use polished rice. They began to make pressing to make clear sake rather than cloudy sake. And the third technique that they developed was pasteurization by heating. And the fourth technique that they developed is using yeast starter or mother culture or seed culture, we can also call, or in Japanese, shubo. So this is the mixture of high concentration of yeast, healthy yeast cell to start main fermentation for sake. This is the fourth technique that temple in Nara developed before. And the last technique that they developed was 3-stage massing or 3-stage addition. So they added all ingredients for main fermentation 3 times separately. I think first day, third day, and fourth day. So they mixed ingredient 3 times in 4 days, not at once. So this is called 3-stage massing. So you So these are 5, 1, 2, 3, 4, 5. And my question is that what is the technique Temple in Nara developed before? What makes long storage or long journey, transportation for sake? It was the, pasteurization. Due to this procedure, due to this technique, we can transport sake from Osaka, Kyoto area to Tokyo, Edo area by ship. So this technique could prevent spoilage of sake. Even we can store sake for longer, or we can transport sake for far from the producing area in Kyoto or Osaka or Kobe area. So pasteurization makes sake. Pasteurization make us to keep sake for longer or to transport sake far distance, long distance. This is another question. This is the slide showing some information. So the energy efficiency, fermentation, O2, small amount of water. So you can find that the energy efficiency to produce energy, for example, by yeast, in the presence of oxygen, is much better than the efficiency of energy production in the absence or under the low oxygen condition. Energy efficiency to produce energy with oxygen, large amount of oxygen, by respiration is about 40%. On the other hand, if we don't have much oxygen in the environment, and organisms like yeast or lactic acid bacteria will make fermentation rather than respiration. But energy efficiency means, energy, what they can obtain from one molecule of glucose. Only 2.3% of energy they can obtain from one molecule of glucose. But if we have large amount of oxygen, and they can make respiration, and more than 40% of energy found in one molecule of glucose can be obtained. So energy efficiency or energy producing efficiency by respiration is much better than that by fermentation. This is the slide showing the location or localization, location within the cell. So this shows the cell. And the glucose enters the cytoplasm through the protein. And it will be degraded, break down to pyruvate. This process is called glycolysis. So glycolysis occurred. in cytoplasmic space, cytoplasm. So you can remember glycolysis from glucose to pyruvate occurred in cytoplasm. And next, pyruvate will enter into the matrix of mitochondria. And TCA cycle occurred. And electron will be transported the membrane of mitochondria. And ATP will be synthesized. So you can memorize, you can remember glycolysis occurred in cytoplasmic space, a cytoplasm of cell, like yeast, or even by ourselves. Our human being also glycolysis is performed in cytoplasmic space, cytoplasm. Yeah. So I think you should be okay with some information regarding the midterm exam. You can just choose 1 out of 5. It will be multiple choice. So shouldn't be much problem. So we can move on to the subject for this afternoon. Share. So I will move on to the slide. Share. This should be. Yeah. This slide shows the enzyme activity or enzyme, what we can find in Koji fungi. May remember this photo showed the Koji fungi grow on the surface of the steamed rice. Usually we use fungi, Koji, yellow-colored Koji for sake making. But these fungi, its color is black, have been using for making distilled sake. Our shochu is the name of distilled sake, especially in the southern part of Japan, like Okinawa Island. The temperature is very high, so sake will be easily spoiled. But this fungi makes a large amount of citrate, acidic compound. It prevents sake from spoilage. It inhibits the growth of contaminated bacteria. But if we use this fungi for making sake, and the taste, aroma, and the balance of sake, quality of sake is not very good. So this type of fungi is specialized to make distilled sake, a shochu. This is the color mutant. We also call white koji. This is a color mutant of this black fungi, black koji fungi. And some people use this type of koji fungi to saccharify starch of rice to make shochu, especially shochu or distilled sake. And some people blend 2 types of fungi to saccharify the starch of steamed rice to make sake. And this fungi is a provider. These provide the enzyme activity, not only for saccharification, like alpha amylase or G amylase that we discussed last week. You may remember we have at least two enzymes for glucosidation. Yeah, this is alpha amylase. So starch will be break down to make maltose. Starch will be degraded to maltose. This is an enzyme called alpha amylase. And the maltose will be digested to glucose. This is the G amylase or alpha glucosidase. So alpha amylase, G amylase. We have at least two genes to encode, one for alpha amylase, another one encode for G amylase. So this is the alpha amylase, G amylase. So if we make ginjo style of sake, ginjo is made by 60% or less than 60% of rice we use to make ginjo sake, which has, ginjo sake has aroma, some floral or fruitiness aroma ginjo sake has. So if we want to make ginjo style of sake with strong aroma and refreshing taste, and we should use koji fungi with high activity of G amylase rather than alpha amylase. So if the koji fungi alpha amylase has more activity than G amylase, and the sake will have less aroma and less taste. So if we want to make more aroma and more taste sake, and we had better choose koji with more G amylase activity than alpha amylase activity. So people try to select the koji fungi, depend on what type of sake they want to make. This is another glucosidase, but it is not very important. This enzyme converts sugar. A 3 molecule of glucose will be linked by this bond, and it will make oligosaccharide. But yeast cannot utilize. So this makes some, delicate taste of sake. This oligosaccharide will be stored because yeast cannot digest it. And sometimes this oligosaccharide will produce delicate taste within sake. But it will not be, this is not very, very important. And these are glucosidase enzyme. And we have also, koji has protease. So koji has enzyme to break down protein to make amino acid. The major protease, one is called acidic protease. The optimal pH for this protease is acidic condition. So that is why people call this protease as acidic protease. This protease randomly hydrolyzes the peptide. But it does not break down protein into amino acid. So this makes some peptide. But it cannot make amino acid. On the other hand, acidic carboxypeptidase, acidic carboxypeptidase. This is another protease. So this also cleave the protein. It makes amino acid as well as peptide. So this makes umami taste by making glutamate. Or this also makes aroma, aromatic compound. Yeast will decompose the product. But this peptidase produced. And long-chain alcohol or ester will be made by yeast. It makes aroma. So this enzyme activity or acidic carboxypeptidase is another important factor to select the koji fungi. It depends on what type of sake brewery want to make. If they want to make sake with more umami or more aromatic compound, and they need to be careful to choose koji fungi with acidic carboxypeptidase activity. And the other enzyme is not very important, tyrosinase, oxidized tyrosine, to form melanin pigment. So if koji fungi has high activity of tyrosinase, and the sake will be made with colored brown. So not very good for sake. So probably we had better choose koji fungi with low activity of tyrosinase. And the sake will not have much color. Lipase. This cleave the lipid to produce glycerin and fatty acid. If we use the small size of rice grain by polishing, and it will not affect very much on the taste or aroma of sake, because from outer layer, lipid or fat localize outer layer of rice grain. So if we polish very much, and we can discard fat or lipid from rice grain. So if we use small size of rice grain to make sake, and this enzyme activity will not affect very much because rice does not have much fat or lipid. So these are major enzyme activity or enzyme what we expect koji to make sake. So koji is a provider or source of enzyme, these enzymes. But we need to control very carefully when and how much this enzyme activity will be expressed by the gene, from the gene, what koji has, what koji fungi has. So I try to arrange to ship sake to your university. So we have, JETRO is the name of organization, Japan External Trade Organization. They have office in Lima. So Japanese Ministry of Education, what they awarded our program, they began to contact to JETRO in Lima to arrange, different style of sake you can taste. I hope they will work efficiently. And we can, or you can try to sake, different style of sake. So I will move on the subject to East. The next subject will be East. Share window. No? People to work on wine made by grape, they do not recognize yeast are very important. Usually they pay more attention on grape itself. But yeast, as you already know, produce long chain alcohol or ester. So yeast are very important materials or ingredient to make sake or even for making wine. So next, some slides I try to explain yeast. So you already know yeast is a eukaryotic organism. So it has nucleus within a cell. But this is a single cell molecular microorganism and classified as members of fungus kingdom. So this is a kind of fungi made by single cell. So this shows the location of budding. They try to divide a single cell to make more cell. Sake yeast. People began to pay more attention since the year of 1917. Okay. So these are number. Japanese government made this number 1, strain number 1, strain number 2. This is just the order. But they began to provide these yeast to the brewery. Year of 96. Masamune is the name of brewery, name of company in Kobe, Hyogo area. Japanese government began to make contest for newly made sake since this time. And they, some sake produced by this brewery is called Sakura Masamune. Sakura means cherry blossom. Sakura Masamune. They made a great, sake. So the sake produced by Sakura Masamune was awarded. So people paid attention. The yeast that they used, and they tried to isolate the yeast strain from their brewery, and they found one strain, and later it's called strain number 1 in year of 1906, and the government began to distribute this strain to other breweries in Japan. And next, 6 years later, 1912, Gekkeikan. Some of you may familiar with this name. Gekkeikan is a name of brewery in Fushimi, Kyoto area. And their sake was awarded at the contest in Japan. And the government tried to isolate the East from their brewery in Gekkeikan, Kyoto. And they isolated one East strain. And the government began to distribute this strain from year of 1912, which was called as the strain number 2. 2 years later, next time is in Hiroshima Prefecture. So Hiroshima sake produced in Hiroshima was awarded. And Seishin is the name of brewery in Hiroshima. Their sake was awarded. So people, Japanese government tried to isolate the East strain, but made Seishin sake. And they began to distribute number 3 East strain to the brewery in all over in Japan from year of 1914. And same thing happened. But number 4 strain of East, we do not know, people do not know exactly this East strain was isolated from where. Some brewery in Hiroshima Prefecture, certain brewery in Hiroshima Prefecture was the origin of the East strain of number 4. But these days, nobody knows which brewery, from which brewery this East was isolated. And it has been distributed since year of 1924. And next, oh, but maybe something wrong with this number 26. I will make sure. Number 5 East strain has been distributed since 1926. Kamotsuru is another famous brewery in Hiroshima Prefecture. So these 5 East strain was, the famous East strain used to be distributed to the many brewery, or most of brewery in Japan, sake brewery. But these days, these are not distributed by Japanese government. So this continues. But still, they have their own East strain, like Sakura Masamune Brewery keep their number 1 East strain, Gekkeikan in Kyoto. They have been keeping number 2 strain. So these days, they try to make revive sake by using number 1 East strain. So this is the sake produced by Sakura Masamune Brewery in Hyogo, Kobe area. It is called Yakimare Kyokai. Long name is number 1 Junmai. So as a commemorative sake, they sometimes make sake by using very, old classic strain. It's called just like number 1 is strain. But they, lost their record how to make sake by using this strain, number 1. So they already lost their protocol how to use this strain. So they, struggled. They have been struggling for a long time to make sake, to make revived sake. But finally, they made it. Polishing rate, 80%. So, it may not have much ginjo aroma, fruity aroma, or floral aroma. So this supposed to be the very traditional sake. It has traditional taste like cereal or style sake is supposed to be. Alcohol level is between 15 and 16%. Sake meter value 16 means it is very dry. This is the comment what they gave me. Rich taste of rice spread throughout the mouth, resulting in a rich, strong, and crisp sake. This is the comment what they gave me as their impression about this sake. So, Gekkei can also try to make very similar, project. So this is a East strain. For a long time, nobody, used. So these are forgotten East strain, but they, Gekkei kan, who isolated this strain more than 100 years ago, but they made sake to celebrate, 100 years since they isolated this strain. So this is the sake what they made by using classic number 2 East strain. Oh, they claim Junmai, it means pure rice. So this sake is a Junmai, no additional distilled alcohol. Daiginjo. Daiginjo means, so this sake was produced by the rice grain, less than 50% of polishing rate. Daiginjo. Polishing ratio 50%. So we can, or they can call this sake as daiginjo. And they did not add any distilled alcohol. So Junmai, they can also put the name of Junmai on the label. So they put here Junmai Daiginjo. Junmai Daiginjo. They can put the word on the label of this sake. This is the comment what they gave me about this sake. Sake made by number 2 East has fruity aroma, such as apple and blueberry, due to its refreshing aftertaste. Roast beef, white fish carpaccio, pizza, and pasta, and even vanilla ice cream pair well with this sake. This is just their comment. I have not tasted or tried. When you have a chance to go to Japan, you may be able to access this type of sake. This is also Suishin, who isolated number 3 East. They also made this type of sake. They put the 3. 3 means, they use number 3 East. They did not they did not send any comment about this sake to me yet, but I will let you know once they will, introduce the impression or character of this sake to me. So the company, originated these East strain. Some company tried to make revived sake by using classic East strain. And more strain of East has been isolated. Number 6, 7, 9. These are the East strain. Still, we can distribute it to the brewery in Japan. They are very strict, and they try to protect these East strain. So they are not allowed to ship to the overseas. But I have been working with the National Institute of Brewing Research in Japan. So they provide this strain to me. So you can, work on these yeast strain to make sake. And also I made beer by using these yeast strain. And we can make very interesting taste of beer by using these sake yeast. So if we can start to work on lab course at your university by using these yeast strain, it should be fun to make sake as well as beer by using these yeast strain. Numbers. Oh, it's already 5 o'clock in Lima, Peru, Arequipa, and 6 o'clock in Bloomington, Indiana. So we can make brief break, 5-minute break. And I will continue. I will resume to discuss about some more yeast strain and characters of yeast strain to make sake. The second half of this afternoon. So Kenny, can we make 5-minute break?
Ok, profesor.
Okay.
Chicos, entonces son las 5:01. 5:06 estamos, por favor, reiniciando nuestra sesión. Muchísimas gracias.
Kenny, can we read you?
Okay, Professor.
Okay. Oh,
Thank you,
let's-
Professor.
Okay, you're welcome. So let's start the second half of this afternoon. So these are another, major yeast variety or yeast strains isolated from Akita, Nagano, Kumamoto. It means not Hyogo, Kobe, not Kyoto, not Hiroshima. So these are three cities we call three capitals of sake production because it has long tradition. Hyogo, Kobe area, Kyoto, Hiroshima. So these are very famous capital of sake production. But you can see these are from 1906 to 1926. But these strains 6, 7, 9, 1935, 46, 53, 91, 2006. It means sake production or is isolated from other area in Japan, not Kyoto, not Kobe, Hyogo, not Hiroshima, Akita, located the northern part of Japan, Nagano, center part of Japan, Kumamoto prefecture, located southern part of Japan, Tohoku, northern part of Japan, Hokuriku, northern part of Japan, Akita, again Akita. So, people pay more attention the very characteristic sake produced in other part of Japan. So it means the people's preference diverse. At the beginning, government control very much. So these East strain, government tried to made the initiative to isolate, but the government tried to award the sake, grade the sake at the competition. But later, people try to began to realize very characteristic sake produced in other part of Japan. For example, the ginjo, you can find the ginjo, word of ginjo, aroma of ginjo, producing ginjo, aroma of ginjo. It means, trend. Popularity of sake is now more ginjo. I mean, people try to find yeast strain to produce aroma of ginjo. So this is the trend of Japanese sake these days. At the beginning, sake with ginjo aroma, like a fruity aroma or floral aroma, people do not like it. They prefer to drink sake with the impression of cereal. I can explain later. Sake has some aroma, and the people used to prefer sake with, cereal or fungi. Impression of cereal, impression of fungi. But later, especially after 1980 or 1990, sake with the aroma of ginjo became very popular. And, people in the world, overseas, also prefer sake with aroma of ginjo, just like wine. So that is why the reason why people try to find the yeast strain to produce more aroma of ginjo. This is the difference between sake made by large size of rice grain compared with sake produced by small size of rice grain by highly polished. So 70% rice means, we discard only 30% of rice grain. So sake produced by the rice with 70 or 60% polishing rate has more body, but less aroma, less fruity aroma, or less floral aroma. But ginjo or daiginjo sake produced by the rice, the polishing rate is less than 50, and we can call daiginjo. Has more fruity aroma or floral aroma, but the body is not very heavy, very light body. So this is the image of sake. Depend on the ingredient of rice in terms of polishing rate. So people used to prefer to drink sake like this. They like more umami. People like more body. People like sake with more sourness, sweetness, or bitterness. But these days, people prefer sake more like this style. Not much body, but people like more fruity aroma, floral aroma. So this is the ginjo aroma. So you can, imagine the trend has been changed from the taste of traditional sake to sake with ginjo aroma, is a fruity aroma, a floral aroma, which was made by the East strain, certain East strain, like, these East. To make ginjo style sake, we need to keep temperature low. So this graph shows the temperature. So y-axis shows the temperature of the fermentation, during the fermentation. X-axis shows the days of fermentation. 普通酒 ordinary sake show, this blue line shows the temperature change when 普通酒 sake is making. 普通酒 is produced by the rice grain, about 70%, or more than 70% of rice grain. The large size of rice grain will be used to make 普通酒 ordinary sake. And the green line shows the temperature change during the fermentation to make ginjo sake by using rice grain. Of the polishing rate is less than 60%. So you can see in the temperature to make ginjo sake lower than 10 degrees Celsius. So we have to keep the temperature very low, between 6 or 5 degrees Celsius to 10 degrees Celsius. And it takes a long time to make ginjo sake, more than 1 month. We need to complete fermentation for ginjo sake. So we have to keep temperature very low, and yeast will produce a ginjo aroma, fruity aroma, or floral aroma. On the other hand, to make futsushu, fermentation will be completed less than 3 weeks. And the temperature is higher than the one to make ginjo sake. 15 degrees Celsius it reaches. So this is the difference. So based on the procedure to make futsushu, yeast will not make much fruity aroma or floral aroma like ginjo aroma. But this sake has more impression of cereal or fungi. So this sake has the taste traditional sake. On the other hand, people in overseas or more people in these days prefer to drink, taste ginjo style of sake by following this procedure. This is the difference how to make two styles of sake, ginjo sake or futsushu, ordinary sake. We'll skip. So this is an image of aroma. Ginjo sake or dai ginjo sake has floral, apple, pineapple, banana, melon, and pear. So we can feel, aroma of pear, melon, pineapple, banana, apple, as well as roses, like flower. If we use the small size of rice grain and a certain strain of yeast strain and fermented at the low temperature, and we can feel this type of impression from ginjo sake. So these are typical aroma of sake. We can find fruitiness. This is a kind of ginjo aroma: apple, pear, banana, melon, lychee, strawberry, citrus. And another aroma profile: alcohol, spice, nuts, alcohol, clove, cinnamon, fenugreek, nuts. This type of profile sake also has. And the grass and the green profile of aroma sake also has: cedar, grass, rose. So these three are major impressions we can feel from ginjo style of sake. And on the other hand, futsushu, ordinary sake, produced by your traditional technique, relatively high temperature with traditional yeast strain by using rice. Polishing rate is about 70% or more. So by using large size of rice grain at the high temperature, we can feel sake, the aroma of cereal or fungi. Rice, we can feel impression of rice from traditional sake, koji, fungi. And also we can feel the characters of honey, dry fruit, brown sugar, and soy sauce. Also, we can feel impression some acidic like vinegar, yogurt, butter, cheese. So this comes from mother sugar. Because traditionally people apply the lactic acid fermentation to make starter. So it has some, yogurt, butter, cheese produced by the lactic acid bacteria. So sake, made by traditional technique, has some impression of the daily milk, yogurt, butter, cheese. So traditional sake has more impression of these three. On the other hand, modern sake, ginjo sake, has more impression of these three. So 普通酒, what I explained earlier, ordinary sake or honjozo sake is produced by the rice that the polishing rate is 70% or more than 70%. Honjozo has this type of impression. On the other hand, modern sake or ginjo style sake has more impression of fruitiness or spiciness or grass and green impression. So I try to explain this has aroma from banana, either amyl acetate or ethyl caproate, which is aroma from apple. So these are compound yeast produced for making sake, to make sake. So this compound also made by the yeast, small volatile compound. This small volatile compound, yeast produced as an ether. The is the composition, ginjo aroma. Origin, this compound is produced by yeast during fermentation. At a high unsaturated fatty acid level, its production is suppressed. But the aroma of apple produced by yeast during fermentation. Some mutant of yeast produce more ethyl caproate. So people try to isolate the mutant strain of yeast, which makes more ethyl caproate, which makes more isoamyl acetate. So this is another trend to study or to develop new yeast strain by mutation. I can explain how to make mutant to produce more isoamyl acetate, to produce more ethyl caproate. So these are, apple, banana. So it means, by using mutant yeast strain, which produce more impression of apple, which produce more impression of banana, people prefer to taste. So these sake with more impression of apple, with more aroma of banana are very popular these days. So I can skip. So this is a slide showing phylogenetic trees of Saccharomyces cerevisiae. This is a taxonomy of yeast strain to produce sake as well as wine. Beer should be ale beer. agave, Mexican agave is the ingredient, raw materials to make tequila. So people try to isolate yeast strain from different, producing area like dairy, and milk, beer, tequila, beer, cocoa. For making cocoa, fermentation process also involve wine, palm wine. Or I explained the palm wine, about palm wine, in the first lecture of this program. Taiwanese. Oak tree. And sake. So sake is located about this area. So it is far from the yeast strain to produce wine or wine yeast. Wine yeast locate 1 to 4. 1 to 4. So these are group of yeast isolated from the wine producing area in Europe. And this is the yeast strain isolated from the sake producing area. And in between, we have yeast strain isolated from beer producing area 11. So this is the yeast isolated from beer production area. So they, have, because they belong to Saccharomyces cerevisiae. All these belong to same, taxonomy. So it has similarity, but still, wine yeast and sake yeast are not close compared with wine yeast and beer yeast. So we can, make, so as I told you earlier, if we use sake yeast to make beer, and the beer has very characteristic taste, and I really enjoyed beer produced by sake yeast. So I also looking forward to make wine by using sake yeast. Or also, we can make good sake or interesting sake by using beer yeast or wine yeast. It should be fun to make, variety of alcohol beverage by using different ingredients and different strain of yeast strain. I can explain some example how people try to develop new yeast strain. These days, most of local government, they have their institution to develop technology for food science. And I try to explain one example which has been performed in Kagawa Prefecture. This is my hometown in the southern part of Japan. And it has a Shodoshima Island, small island. This is the island. Japanese government succeeded to plant the olive tree in Japan. Government, more than 100 years ago, Japanese government tried to grow olive plant in Japan. And they tried to plant 3 different locations to olive, for olive growth. And this island, small island, was an only location Japanese government succeeded. So olives was survived during the winter. So it means this has very, comfortable climate. The winter, it is not very severe during the winter. So this is the island. The Kagawa Prefecture local government tried to isolate the unique yeast strain from olive tree. So this is the olive tree found in this island. It's called Shodoshima Island. So they tried to isolate the yeast strain from flower or fruit or leaves. And they, isolate and analyze the yeast strain, but they isolated from different part of plant, olive tree. At the beginning, we need to screen by TTC test. So red color colony with red color, strong red color, show the high activity to produce alcohol. So first we need to select yeast strain showing very bright, strong red color. So we discard, we need to ignore the colony with pale pink or white colored colony. And they concentrated the colony strain, which shows very strong red color. And next, they tested the activity for fermentation, and they selected yeast strain, which has the high activity for fermentation. And next, they also selected Saccharomyces cerevisiae by PCR to amplify certain DNA sequence. So these strains show in size of band, small size of PCR product were discarded. They gave up. Even these strains showed high activity for fermentation, but to produce alcohol. But they keep only the yeast strain showing high activity to produce alcohol and Saccharomyces cerevisiae. So they keep these strain. And this is the data of their evaluation. So these are 5, maybe 6 different candidates they selected, they keep. But you can see alcohol level after fermentation is not very high. Usually sake has more than 15% of alcohol, but they produce less than 10% of alcohol by using these strains, but they selected. And also, only these 2 out of 6 strains were Saccharomyces cerevisiae. So they keep these 2 strains. So these are not Saccharomyces cerevisiae candida, and these are name of taxonomy, but they isolated from, these are isolated from flower or olive plant. These were the yeast strain isolated from fruit of olive. So they keep these two strains of Saccharomyces cerevisiae, but they did not have confidence, these two, because, alcohol level they can produce was not high enough to compete with the other bacteria which caused contamination. They try to isolate the strain which produce more level of alcohol, by this selection. Maybe this is screening or selection. So they try to transfer their candidate two strains on the 12% alcohol level. Now, it means they try to screen the yeast strain, yeast cell, which can be survived at a high level of alcohol. Next, they pick up the colony from the agar plate with 12% of alcohol, and they transfer to the another plate with 15% of alcohol, and they pick up colony grown on this plate, and they transfer to the agar plate with 18% of alcohol. And finally, they obtained two strain from 04 and 05, one from each. OY04 can be survived or can produce 15.6% of alcohol. OY05 can produce 13% of alcohol. So these two their original strain, OY04, can produce only 9.4, but after screening, it was improved to 15.6 from 9.4% of alcohol production. And same thing happened on the OY05, 13%. It was improved from 8.2% of alcohol. So by this type of screening, they screened these two yeast strain. So this is a control. 9. So this is the strain of number 9, as I told you before. Oh, this is a strain called number 9, isolated in 1953 in Kumamoto Prefecture in Kyushu Island. It makes sake, bibicus flavor, and characteristic aroma of ginjo. So they use this strain as a control. So this strain, number 9, produce more than 19% of alcohol level. And you can see SMV number. So I will explain this later, but if this is minus 14, so sake supposed to be very sweet. If this number is getting bigger, like a plus 1, 5, 10, and sake will be dry. So this sake made by this condition supposed to be very, sweet sake. Acidity and amino acid. So this is the data obtained by small-scale test brewing by using 200 grams of rice, not much. So we can make this type of test brewing by using 50 milliliter medium bottle. So if we have a chance to make lab course in Arequipa, and we can make test brewing, just like this by using rice, koji rice, water, and yeast. So these are 4 major ingredients to make sake: rice, koji, yeast, and water. So just we can mix these 4 ingredients in the medium bottle of 500 milliliter or 1 liter of bottle. And they evaluated the product and alcohol level by using control number 9 strain, which produced more than 20% of alcohol from small-scale test brewing. And candidate, 2 strain of candidate, OY4, OY5, and they got a good data number. About 90% of alcohol was produced by using OY4 strain, but OY5 less than 50. This number is very constant, I think. 15, 30, small scale. And they try to make small scale brewing several times. 18, 14, 19, 40. So probably, they decided to use OY4 strain by this data. Test brew. SMB is now improved. 3947. Sake produced by OY4 is not dry compared with sake produced by number 9 strain. But still this number is not very bad, very good. We can enjoy sake with SMB of 3947. Acidity. It seems to be more acidic compared with sake produced by KY9. So it has more acidity. Amino acid level about the same. So it may have about the same level of umami of sake produced by control with number 9 yeast strain. So this number can be satisfied by many people. So that is, and so based on this data, they, so this showed the photo showing small-scale test brewing with bucket. So very easy, just mix with water, steamed rice, koji, and yeast. This shows this is a large scale of test brewing. Not this type, but they try to scale up by using number 4, KY number 4 is strain. And this is a 4 company these days are making sake by using the yeast strain, but they isolated from olive fruit. So this is the one story, how people try to isolate new yeast strain. So what type of evaluation we have to make? We need to be clear. This is the one example. But people try to develop a strain of yeast. So this is, they want to use the word of olive because this is the, olive is the characteristic product produced in Kagawa Prefecture. So they try to want to, like olive sake or olive. But more people try to develop new yeast strain, which produce more aroma of banana, more aroma of apple. So rest of time, I try to explain how to make yeast strain, which produce more aroma of banana, more aroma of apple. Very complicated. So first, I try to explain the mechanism. But we can make yeast strain to produce more aroma of banana. There is the isoamyl acetate. This is the chemical compound, volatile compound, which has aroma of banana. So we need to make yeast strain, which makes more isoamyl acetate. This compound will be produced from glucose. So this slide shows the pathway, how to make isoamyl acetate from glucose. So now you are familiar with this metabolism. Glucose from pyruvate. You may remember the word of glycolysis. Glycolysis occurred in the cytosol of the cell. So this is the, one question of midterm exam, where glycolysis will be occur. And it will be in the cytosol. We don't need any oxygen from glucose to pyruvate. And it will be converted to, isobarrel This compound itself does not have good aroma. But when this compound will be converted to isoamyl alcohol, it has some aroma. So first, people try to improve the amount of isoamyl alcohol first by manipulating a metabolic pathway. Usually, leucine is an amino acid. This makes feedback inhibition. So if the cell makes more leucine, it will try to inhibit this enzyme as a negative feedback. And this, this metabolism will be stopped or will be inhibited. And if negative feedback occurred, isoamyl alcohol will not be made much. So first, people try to release this negative feedback by making mutant of this enzyme, within this enzyme, alpha isopropyl synthase. It means even we have lots of leucine, but leucine cannot bind to this enzyme. So negative feedback will not be made And this, metabolism will, makes more and more. And it means more isoamyl alcohol will be made. So first, people try to isolate mutant by isolating resistance strain against leucine analog 5', 5', 5', trifluoroleucine. So this is a leucine analog. So if we have resistance mutant against this leucine analog, it means, leucine or TFL cannot bind or cannot inhibit enzyme activity of alpha isopropanol synthase. So it means we will make, or cell will make more isoamyl alcohol. So this is the first step people try to isolate. And by making this mutation or isolation, I think this is performed by Hiroshima Prefecture, local government of Hiroshima Prefecture try to make it, and they obtain mutant, which produce 5.2 times higher level of isoamyl alcohol by selecting mutant on alpha isopropyl malate synthase. But we need to have more isoamyl acetate. Isoamyl alcohol has the aroma of very alcoholic aroma, but we need to have the aroma of banana, which was provided by isoamyl acetate. So we have to improve the to make isoamyl acetate from isoamyl alcohol. So next, they made another mutation. Detoxified by esterification. Yeah, this enzyme will isoamyl acetate. In order to make isoamyl acetate, they use this type of chemicals, which is called fleginolone. It is a toxic for yeast. But this will be detoxified by ATF2P gene product enzyme, which is called alcohol acetyltransferase. So they try to screen the yeast strain, which produce more enzyme, which convert from isoamyl alcohol to isoamyl acetate. So they try to screen the yeast, which produce more enzyme, which convert from here to here by selecting the resistance mutants against this compound, because ATF2 gene product esterify this product, which makes detoxification of this compound. So by isolating the mutant, which has resistance against this enzyme, it's supposed to make more gene enzyme activity of ATF2B. And they found that, they obtained double mutant. One mutation occurred in this gene, and another mutation probably somewhere in here. And as a result, they have obtained the yeast strain, which produce more isoamyl acetate, which means the yeast which can produce more aroma of banana or more aroma of ginjo. So this is the one story. People in Hiroshima Prefecture try to isolate yeast strain, producing more aroma of banana by manipulating metabolic pathway to synthesize isoamyl acetate from glucose. You don't have to memorize these, metabolic pathway, but you can just understand the trend. People try to work on to make new strain of yeast. So this is aroma of banana. It's one keyword. These days, people try to work on. So next, here's the aroma of apple. Ethyl caproate makes the aroma of apple. So this shows the metabolic pathway to make ethyl caproate. So people try to make mutant on enzyme called fatty acid synthase by selecting cerulenin resistance gene of yeast. So they try to isolate the strain which has mutation within the gene of FAS2 gene. What is this? This makes the fatty acid. Okay. So this is the mutant which has resistance against the cerulenin gene. So it means right side of the bar, each amount of each fatty acid, mutant has less amount of long chain fatty acid, 18, 16, but it has more short chain fatty acid. So if you take a look at, not very much, but see carbon 6, fatty acid made by carbon 6, cerulenin mutant, resistant mutant has more C6 fatty acid than wild types yeast. And C8 also, cerulenin resistance mutant makes more. C10. So it means small size of fatty acid will be produced within the cerulenin resistant mutant cell because the long chain fatty acid production was inhibited within cerulenin resistant mutant. And as a result, ethyl caproate, this is a kind of part of fatty acid. So that's very short, small size of fatty acid production improvement. As a result, ethyl caproate production also improved in these mutant cells. Not only small size of fatty acid production, but also ethyl caproate production was improved within the cell, which has mutation, which makes resistance against cerulenin, anti-fungi chemicals. So I try to explain a little more about this in next week. So I will stop at this slide, and next week I try to complete this story and water, and I try to introduce some character of sake, but I explain a little this afternoon. So Kenny, can I stop here this afternoon?
Okay, professor. Thank you
Okay.
for this session.
Thank you very much.
Chicos, antes de terminar, solamente recordarles que ya tenemos el